This doesn't work. Does it work? Yeah. Well, thank you very much. Thank you very much, and thank you very much for Nick and the organizers to invite me to give this talk. And I know that it's very early in the morning, and it's a nice, cozy meeting. I'd like to talk about peptide delivery. Our group in the University of Queensland working on drug delivery in general, including gene, peptide, and vaccine delivery. Now I will concentrate on the vaccine delivery side of our work. I start my talk with the most important slide, of course, in this whole lecture. This is my research group. I work with very nice people. If you were here yesterday, several of them gave a small talk about our research. We have about 6 postdocs, 30 PhD students, and several honors and master's students in the group. So obviously their excellent work lets me do the talks and apply for different grants. We are working on vaccine delivery in a big group funded by NHMRC in the third 5-year term of the program grant. And there is 4 immunologists, 3 clinicians, and I'm a medicinal chemist specialized on drug delivery. Vaccination, as you know, the biggest blessing of the human race, and it's responsible for 15 to 20 years increase in life expectancy. And we know that it's given most of the time in childhood. It has to be extremely safe. And the very first vaccination, which was mass administered, happened 200 years ago, and they realized, as you very well know, that the cowpox serum, cowpox-infected cow serum protects against smallpox, and that was the beginning of the new era. But this picture was taken several hundred years ago, or perhaps even thousands. So the vaccination is known for a very, very long time. It's the fastest growing field in the pharmaceutical industry. And if you just see this date, 1798, when the first smallpox vaccination was given, roughly about 10 years after, this painting appeared that shows the first public kind of fear and against the vaccination. I don't know if it's a coincidence or it's really happened, but this happened roughly about 10, 15, 20 years after the introduction of the vaccination. But obviously today is really not much difference. Now in 2014, if you go to the press, you see all those articles, because nowadays, especially well-educated families just refuse to vaccinate their child. Obviously this might have really tragical consequences. And now obviously a big campaign started against it, and hopefully this whole fear against vaccination will be changed. So the successful and cost-effective health invention really controls many infectious diseases, and there are problems with it. If you see this picture again, it's 200 years ago, when they took the cow serum and injected to people directly, the vaccine production is not much changed these days. It still use eggs, still use horses, so still use biological system to produce vaccine. Then we use whole microbes or part of the microbes, and it sometimes really represents a problem. Risk of infection, autoimmune responses, and limited shelf life. So the solution could be synthetic peptide-based vaccine. What the synthetic peptide-based vaccine is, here's the microorganism, and there are several carbohydrates and protein on the surface of that microorganism which are responsible for the immune response. If you just use one of the proteins from this microorganism and with the right configuration, it generates antibody which will recognize the whole microorganism. Go further, even part of that protein, if you go for antibody response, you have to have a small portion which has exactly the same secondary structure of peptide, or if you go to T-cell response, it doesn't really matter. The structure, you can use that small part, and the antibody generated against the small part of protein or peptide will recognize the microorganism as well, and it will do its job. But although it's very advantageous. have advantages because remove unnecessary components, so decreases the risk of autoimmune disease, non-infectious, and customizable. But there are problems with it. By removing the unnecessary component for the vaccine, for the antibody design, we remove the danger signal. So the body won't recognize the small peptide on its own. Needs strong adjuvant. And all the adjuvants, even the recently approved from Steve Reed, I think, is still toxic. In case of peptide, the small molecular weight limits the capacity to induce immune response, so we have to be coupled to a big carrier protein. And furthermore, we need a T helper epitope if we want to have efficacy in an outbred population. So there are several attempts to overcome this problem and put this more peptide sequence into a construct which is recognized by the immune system. And there is a polymeric approach when you have a peptide sequence, you put an acrylic acid in there and they polymerize, so you have a carbon chain, and those epitopes, one, same or different, you just hang on this polymer chain, or you design an amphoteric peptide molecules which form and rearranges, give you a secondary structure in dendritic way, or you put them on polymer surface. And in the world, many research groups started to work on lipid modification of those peptide sequences, because the lipid modification of a hydrophilic peptide makes the compound amphoteric, and gives you the possibility to get the rearranged big nanoparticles. For example, Helen Rasmussen in France put this idea together that he conjugated peptides with single lipids and used a mixture of peptides. Her granting clinical phase. Günther Jung introduced the PAM3 cis system. PAM3 cis is a bacterial lipid. You put one copy of peptide antigen on that PAM3 cis, and it will be immunogenic without adjuvant. It was Günther Jung, but in Australia, David Jackson is working on influenza vaccine, using the same idea, PAM2 with 2 lipids or PAM3 with 3 lipids. And it's really great because it can be administered mucosally if it's a self-adjuvanting. By the way, there is no human approved mucosal adjuvant exists. So self-adjuvating compound opens the way towards the mucosal immunization, can generate cytotoxic T-cell response, increase the stability of the compound, and it can be highly characterized because you know exactly what do you make, and it's of course safe. There was another scientist, Jimmy Tang, who introduced about 20 years ago the MAP system, he called this multiple antigenic peptide system, when again he used a lysine. Lysine has on-carboxylic acid 2 amino functions. If you make 2 layers or 3 layers, you can multiply the amino functions on the surface. And you put the same peptide on those amino functions, so the same peptide will be basically on the surface of a dendrimer. Now I saw those ideas, 2 ideas, Jimmy Tang's and Günther Jung's ideas, and I thought we should put them together. And we built up a system where we use a very special lipid, so-called lipoamino acids. It's a very simple amino acid with a normal alpha amino acid on one end, and a long alkyl chain in the other end. You can increase or decrease the length of the alkyl chain. You can make dimers or oligomers of this compound since they are amino acids. Or you can put a spacer between the lipoamino acids, and it's extremely important because it makes the system highly engineerable. So with this system, we mimicked the PAM3 and PAM2 cis special arrangement to deliver in certain positions. You can do the whole thing in solid phase, put lysines, and put peptides on the surface. Now, fortunately, this lysine can be also very productive, so the 2 amino function can be both FMO productive, and with small trick, you can put different peptides on the surface. So if you do a BOC-FMO production, D-BOC it put one peptide on, then D-FMO put another peptide on, or instead of the second one you put another BOC-FMO glycine, you can build several peptide components. So I show how we use this theory using a group A streptococcus model, which is a very, very dangerous, although almost all of us get an infection during the lifetime. There are some form of this group A streptococcus which is extremely deadly, and in Southeast Asia, mainly in the Australian Aboriginal population, is a very, very common disease. And people who get an infectious invasive nuclear step infection, the life expectancy is about 24 years. So it's extremely important to fight against this disease. But it's not that easy, because again, if you use a whole microorganism or one M protein on the surface of this microorganism to generate antibody, antibody will be generated against a relatively conserved C region and very highly variable N region, and the middle part as well, which would be okay. Unfortunately, antibody generated against this middle region of this M protein, which is again highly variable, the C terminal is conserved, it cross-reacted the human heart, causing rheumatic fever and rheumatic heart disease, and obviously this is a deadly disease. So the idea, this is a perfect candidate to make this small peptide-based vaccine design, and we use in the relatively conserved, 70% conserved C region protein or peptide, about 24 amino acid peptide, and it works. It generates antibody beautifully. Only problem with it that this antigen, we generated this antibody generated by this antigen, has to recognize this part, so it has to go deep down in the cell surface, so it's not that protective. If you generate antibody with this end of this peptide, you have to generate many, because there is 140 different cellulas, but we chose 7 very virulent cellulas from Australia and used all those sequences together. And now, as I said, our system is great because you can put different peptide epitopes on this polylysin system, and we will build this system, and with our great satisfaction, this compound, which uses one conserved and 3 variable regions of the compound, is highly productive, give you an antibody response, epitope-specific antibody response with and without an adjuvant. And since it's a self-actuating system, you can use it mucosally. You can use it nasally or orally, and indeed, I don't have to talk about the advantages of the mucosal immunization. It's obvious if you see the comparative the injection. And fortunately, both nasally and orally, the system gave you an epitope-specific antibody response without the use of any adjuvant. And furthermore, what is very important is not only gave antibody response, but this antibody response was protective, and it shows that after intraperitoneal or nasal challenge later, the compound is 100% protective. Now, we synthesize this step by step, but now we have techniques, we make the sequences separately, and with just chemical conjugation or ligation or pre-traction, put them together, so we can make highly pure compounds. So this is now our second generation. Our first compound is in clinical 1 phase, and this is the second generation when you have 2 constructs. One of them contains 1 conserved 3 variable, and the other one contains 4 variable. Using the mixture of that 2 contains all the 7 highly virulent antigen. But now I'm a chemical engineer, I'm not an immunologist, but started to go to immunological conferences and I meet people who actually put this textbook model, you see this in the textbooks, how to generate an antibody response or a T-cell response. But unfortunately, there is no method how to design an antigen which actually will generate, will direct the system towards antibody production. And particularly if you want to make an IgA production, for example, in infectious diseases, IgAs on the mucous surfaces, you just don't know how to design an antigen. And we have some idea of this, the T epitope, but we cannot just generate a T cell response. So we started to work on this system to find out the mechanism, what is responsible to direct the system this way or this way. And the design compound now, because it is method, and you can put together the different peptide sequences in different configuration. We just use a structure activity study on the adjuvant state. So we put those systems together, and we use a T helper epitope now to be able to use any kind of mouse in the experiment. And this is the conserved region. And then we put this in different configuration. Here's the adjuvant, and both peptides, and the helper and the conserved, coupled through the C terminus. This is one of the C terminus, other N terminus, and vice versa. And very big surprise, there's a huge difference in the antibody presentation. It's a logarithmic scale, so every time you have a 10 times jump. So it really does matter a lot. How do you put these whole things together? So we decided to try to examine what's the reason for that. So we use a simple compound now against, this is the model that I showed you, this was the best, which gave the highest immune response, and started to examine that what is responsible for the IgA production. And we started to change the length of the alkyl chain. Really interestingly, it came out only one compound, the highest molecular weight, the longest alkyl chain, 16 carbon long alkyl chain compound gave you IgA response, all gave you beautiful IgG response, but only one gave the IgA response. Now we started to work with that compound, now with the C16 lipid, put them in different configuration, and it does matter which configuration you put it together, because again, we were really lucky in the beginning that the two C-terminal peptide containing compound gave you IgA response, all gave beautiful IgG response, but only this one gave you IgA response, the other two didn't. We started to change now the T helper epitope, and it really makes again a big difference because only that P25 T helper epitope gave you the IgA response. So in the end, we started to examine something, and this is obviously one of the main area of our research now, but we didn't get a step closer, because if you change something which you think works because exactly the same chemical structure and everything, it just didn't. And we started to use now this compound to put together the same T helper epitope and the different sequences now one after the other, and examine its activity in the immune system. And really interestingly, if you put one sequence together with the P24, it gave you a certain immune response. If you put the same sequence with another one, it gave you a much higher immune response, epitope-specific immune response. And the same, the other one, if you put this together, just one peptide sequence, one immune response, and a very small increase when you use the two together. What's the reason? And these compounds are all alpha-teric compounds, hydrophilic head, hydrophobic tail, and can form secondary structures depending on the nature of the lipid and the nature of the peptide, different size. And we started to examine now the size different of those nanoparticles. I didn't really care too much about the formulation of those compounds, but now we know it's extremely important how you present that antigen, what kind of formulation system, what kind of nano system you administer the compound in the body. And interestingly, the compound which gave you the lowest immune response gave you a 100 nanometer size particle size, and which gave you higher immune response. The highest one gave you about 10 nanometer particle size. And obviously now the size is extremely important. We started to check the antigen uptake, antigen presenting cell uptake of the compounds in dendritic cells, in macrophages, and now for our not big surprise, the compound which gave you the lowest immune response were taken up the lowest ratio on those dendritic cells. So the size is very important. So we started to examine the size. One of our PhD students, Nirman, started to work now on different nano-size systems using now liposomes. So the idea is, he was talking about it yesterday, so I don't spend too much time on it. You use this lipopeptide I just showed before and normal cationic phospholipids, liposome-forming raw materials, put this together, and to protect the peptide epitope on the surface, we cover them to positively and negatively charged carbohydrate polymers. And so the idea is that those carbohydrate cover on those liposomes will protect the peptide until it goes through the stomach and will be released in the GI tract. And you see the size of the compound is slightly increasing as you put the different layers on it, and the uptake on the antigen-presenting uptake of the compounds are all very good. And the antibody production after immunization, I said I don't want to spend too much time on it since Neil Mark was talking about yesterday. The antigen generation, IgA generation was really good on the double and triple layered peptides. These sizes are bigger than our original size was. And the size it shows it's about 200 nanometer particle size, but the antibody IgA antibody presentation is really, really nice and good. So we again, we examine further that how important the size is to direct the antibody presentation of the compound. As you see, there was IgG presentation, and these are the double and triple layer compound gave you beautiful IgA presentation. And they cause long time memory response. I don't know exactly the time, it is about 5 months, or how long, 5 or 6 months time, it will give you beautiful IgA response on this compound. So with this one, as far as I know, this is the first study which uses polyelectrolyte covered delivery system to generate antibody response, and obviously we carry on examining this one. So using the same system is not only good to generate antibody against infectious diseases, the system is really good to generate antibody basically anything, small peptides, carbohydrates, and everything. And we are using now in another model to treat prostate cancer and to control fertility. And the idea is that we have this luteinizing hormone releasing hormone, which in the brain acts on the pituitary cells, its receptors, and our body releases it in a pulsatile fashion. As it releases the pulsatile fashion, after go to the receptor, it will release luteinizing hormone and follicle stimulating hormone, which is responsible now for estrogen and testosterone production, and through that it controls the size and activity of our sexual organs. And basically it can be that if now we have an orally available compound, LHR is not available orally, of course, orally available compound, which basically can be administered continuously. Continuous action of this compound, of the LHR on the brain, releases initially a large amount of sex hormones, but with negative feedback, it basically decreases the number of receptors, and the LHR FSH production will be lowered. Alternatively, if you can generate an immune response about our own released LHRH, especially after you decrease the receptors, then it really releases much less sex hormones, and the size and the activity of the organs will be decreased, which is a very, very bad news for a healthy individual, but if you have prostate cancer, obviously it's much better than operation. So we administer the compound. We modify the compound with those lipoamino acids and carbohydrates. I didn't talk too much about just the chemical modification of those compounds, but you can really increase the peptide half-life with lipid conjugation, but obviously solubility goes down. So lipid and sugar conjugation resulted in a compound now when the half-life was much, much higher, and the cell uptake of the compound was much bigger. And I don't want to talk about it too much, but this special glutamic acid, lipoamino acid bond is a product type bond, so it releases the compound in certain places of the body. So you can use this really to control the level of the FSH in this case. You see that some compounds are really decreased the produced FSH. But we made different construct in exactly the same fashion I showed you before for the gas. So we use this luteinizing hormone, releasing hormone with a special epitope to generate antibody against LHRH. And we use different carbohydrate constructs, not only LHR, not only polylysine, but we use carbohydrates and polymers as a core. And indeed, if you administer those compounds to mice, you see a beautiful anti-LHRH antibody production. So we decided to go for big animals. Now we found a very special helper epitope for rams. And we did an experiment when checked the antibody production against LHRH, and this is really beautiful. But more importantly, if you see the size of the testicles of the animal comparing with the control, it really goes down. I have some pictures, but I would advise not to show it. But these will be what we aim to do now, make it orally available and perhaps control feral animal population. And in the end, of course, I have to thank the awarding body. The drug and vaccine delivery research is well funded, fortunately, in Australia, both by the NHMRC and the ARC. and we get funding for the clinical trial for the NIH as well. And thank you very much for your attention.
Thank you very much, Professor Toth, for a very interesting approach to vaccine production. Are there any questions? And Bailey?
There's been a number of innate lymphoid cell subsets discovered recently that respond to a variety of different lipid antigens. But I just wonder if you know whether your constructs actually activate any of these cell subsets, or whether that might be part of the self-adjuvanting activity that you've noticed.
That's a very interesting question, and it's very relevant because we know that our RCP system acts on toll-like receptor 2, 4, and 7, and now we are exhibiting the 17 as well. Yes, I can say yes, we know that. But we have polymeric adjuvant system and we have fluorinated adjuvant system as well, and none of them acts on any of the toll-like receptors. So there are systems which we don't know what causes the adjuvant activity. But the RCP system that I showed you with the lipid works exactly the same way as the PUMP3 system, PUMP2 system.
Any other question? In my mind, the autoimmune effects of group A streptococcal diseases has diminished. And I wonder whether you can envision how to rule out that you will evoke an autoimmune response using your approach.
Yes, you're right, and as I told in my talk, probably didn't spend enough time in it. If you immunize against the whole microorganism or the whole protein, obviously you will have autoimmune response because the antibody generated in the middle part of that protein, irrelevant of the cell wires, that causes the human heart. That's an autoimmune response. But if you choose sequences which they don't cause autoimmune response in the N-terminal, and we know that the C-terminal, C region, consent region wouldn't cause wouldn't cause constructs. That's why these constructs will not cause autoimmune response.
Very good. Thank you very much.